Welcome, everybody. Thank you for joining our panel, How We Responded to the Pandemic. My name is Isabel Bjork, and I'm Executive Director of the Genomics Institute here at UC Santa Cruz. I'd like to give you a little bit of background on the Genomics Institute and then tell you a little bit about our panelists, and then we'll get going. To begin, though, I just want you to know that this is being recorded, so you will have an opportunity to see this again later or distribute it as you like. And also, please note that there is closed caption on here, so if you press the CC, you can get that. And then finally, know that we said that these would be lightning talks from our panelists. So we'll have short talks from everybody. But you also have an opportunity to ask questions. And actually, most of this session is to be able to give those questions to you. So, or respond to those questions for you. We do have questions as well. Please put those questions in the Q&A section, and we will be as responsive as possible to you. All right, so on to the Genomics Institute. I'm really honored to be able to do this panel for Alumni Week because the last 14 months, the Genomics Institute has done an incredible amount of work to respond to the pandemic, and it's been a passion of all of ours. And the panelists that are here today are researchers who have given their time, their energy to really changing the course of the pandemic. So just some background on us. The Genomics Institute started in probably its core action was in 2000 when David Housler, who is now our scientific director, and the UCSC Genome Bioinformatics Group, which was a bunch of very dedicated researchers, some of whom are still with us today, launched the first working draft of the human genome on the internet. And that guaranteed that our genetic code would remain in the public domain forever. And so they subsequently developed the Genome Browser, which is a free interactive web-based microscope that allows you to view any aspect of the human genome at any scale. And the browser supports scientific research worldwide and has served more than 5 billion to date. From this turned into the UCSC Genomics Institute. We now have about 110 staff and researchers on board. We have over 25 affiliated faculty, which are brilliant professors from across the campus. And our overarching goal is to develop and share infrastructure that supports research in the clinical application of precision medicine, human development, evolution, environmental development, and other active areas of research that relate to genomics. So with that, what we've done during this pandemic is really focus on the development of the virus. And that is in a variety of ways, from supporting an incredible effort on this campus to test the virus, to emerging into an effort to sequence the virus and give information out to the public, to health services around the sequences of the virus and what variants are existing in our community and beyond. I will introduce all the panelists before they speak, but I do want you to know now they are all brilliant researchers, and Jeremy Sanford is here to talk about the COVID testing lab that was created on campus that he spearheaded, and to talk about why testing matters so much. Beth Shapiro is here to talk about why sequencing matters and why she has taken her time voluntarily to spend sequencing the virus on behalf of our efforts here. Miten Jain is here to talk about why Nanocore Sequencing is so valuable for the COVID response. And Russ Corbett Dedic is here to talk about why we need to share this information broadly, how we do that, and the bioinformatics of it. And David will talk about why we are now moving towards creating a sequencing hub, which will emerge into an infectious disease center, and why this really matters not only for this pandemic, but for any public health issues that occur in the future with regard to pathogens, so that we can be on top of it next round, should it come. With that, I would like to introduce Jeremy Sanford. So Jeremy is a professor of molecular cell and developmental biology. His work attempts to dissect the roles of RNA binding proteins in mammalian gene expression. So with this background, he was uniquely placed to lead the effort to bring a COVID testing lab to this campus. And that's important because at the time that the lab was started and the ideas for the lab came about as early as February of last year, and it started testing on May 2nd. There was very little testing in the community and there was very little testing in this nation. And so Jeremy and colleagues really pushed to bring the testing onto campus. We do not have a hospital at UC Santa Cruz, so what he did was really extraordinary. I would also like to say that Jeremy is here, even though today he is celebrating his birthday. And so we thank you very much, Jeremy, for that. And with that, I'm turning it over to you.
Thank you, Isabel. I'm going to share my screen and just show a couple slides here. Oops, I just crashed, I think. I'm not going to share my screen. I'm just going to, for now, I think, just talk about, I'm just audibling here. It's reconnecting. Should be reconnected, or maybe not. Okay, so anyways, what I wanted to do is show you a couple slides about how we created a high throughput COVID testing laboratory on campus in about 6 weeks. And, you know, really the goals of this project were to help our community increase high capacity testing with a really quick turnaround time. And we knew that this was important because the faster you can return results to people, the easier it is to break transmission chains. And so our goal with establishing a high throughput lab was that we wanted to be able to both serve clinical providers in our community, and particularly the safety net healthcare providers, and also establish a testing program for our campus. And that would require the ability to run thousands and thousands of tests per day. We identified really, 5 or 6 key roadblocks that we had to overcome immediately. Those included, having to develop a cost-effective, rapid testing program that have completely avoided supply chain bottlenecks. We also knew that we wanted to run the gold standard in molecular diagnostic testing. So this is the RT-qPCR based molecular diagnostic. We wanted to make sure that we could work with samples that could be collected as simply as possible, meaning minimizing PPE requirements for the healthcare providers and minimizing pain and suffering for the participant that's being tested. And then we wanted to develop really automated paperless approaches for tracking samples, accessioning samples, reporting results, and of course, processing all of our samples. And the last big challenge we had to do in order to hit a high throughput scale of 7,000 to 8,000 tests a day was to be able to develop a pooled testing paradigm. So we were successful in overcoming all of those. And if my internet on my computer hadn't just crashed, I would have showed you a slide here that was really interesting. It says, an unexpected collaboration in the Santa Cruz Mountains. And this collaboration that occurred was between an entrepreneur and engineer named Joe Ben Beaver, who is the founder and CEO of Joby Aviation, which is an aviation company in the Santa Cruz Mountains building electric planes. But in a previous life, he worked on the Human Genome Project as an automation engineer. And when he finished with that project, he launched a robotics company called Velocity 11, which made a particular type of robot that we coincidentally had just bought for the Molecular Diagnostics Lab. When he caught wind of the fact that we were setting up a lab, he came down and immediately began helping us to put the robot into practice, establish a protocol, and really think about how to expand COVID testing. And together, we hit on a number of solutions that allowed us to completely blow past these barriers. It was a remarkable collaboration, and it's still going today. The last slide that I would show you, it really shows the impact of the Molecular Diagnostics Lab so far. Since May 1st, we've run an astonishing 105,000 tests. More than 25,000 were for our local community healthcare providers. So these would be safety net healthcare providers like Salud Para la Gente and the Santa Cruz Community Health Clinic. We identified more than 3,000 positive cases, all of which were reported back in less than 24 hours. So we're really able to interfere with those transmission chains. Those folks were hopefully isolated and then transmission blocked. On campus, there's a remarkable number that you can see if you look at our dashboard, which says the number of cases person-to-person transmission on campus, and that number is 0. We've done an amazing job at building a program that can test everyone on campus at least twice per week. And this really yielded fruit when the students returned from the winter break in January, where we detected about 70 new cases on campus. They were all immediately isolated, put into quarantine, and then basically there were very few positives after that point. So we were able to do, I think, a terrific job as a group, helping to keep the campus safe from the spread of COVID. The last slide is just a massive list of people. I mean, while I may have been essentially a cheerleader driving this on, there was a huge team of scientists, administrators, policy makers, everyone working together, including all of our patient healthcare provider groups, in order to make this project go forward and be as impactful as it was. So with that, I just want to say thanks, and sorry about the internet dropping off of my computer and not being able to share slides or my video on my phone, but here I am. And you guys, thanks very much, and enjoy the rest of the panel.
Thank you so much, Jeremy. Jeremy has warned us ahead of time. He's off to celebrate his birthday. So enjoy your afternoon, Jeremy, and thank you again. One of the wonderful things about the testing lab is that it has given us not only an opportunity to serve our community, both in terms of our campus here and protect our campus population, but also the community population. So we partner with Salud Para la Gente and with Santa Cruz Community Health, with Encompass, with the jail, and with the county public health to support testing here in Santa Cruz. And the positive cases are then sent to sequencing. So Beth Shapiro is here to speak about the sequencing process. And Beth is a professor of physical and biological in the Physical and Biological Sciences Division in the Ecology and Evolutionary Biology Department. She's also a Howard Hughes Medical Institute investigator and a Howard Hughes Medical Institute professor, and she is Associate Director of the Genomics Institute. Her research aims to better understand how populations and species change through time, in particular in response to environmental and other changes in their habitat. So Beth, with that, I'm turning it over to you. Thank you.
All right, thanks, Isabel. So I'm actually the newest among us to this game here. Jeremy and many others had gotten this testing facility up and running some time ago, and they were doing a tremendous service, obviously, to the campus community and also to the county and more broadly. But it became apparent as the pandemic went on that the way that we were going to be able to get a real handle on cases as they move throughout the community would be to be able to do more sophisticated types of tracking. Of course, if someone is identified as positive, you can isolate that person and make sure they don't have any contacts. But if you want to know if there's a cluster of transmission, so you can get to the root of what that cluster is, or understand maybe travel pathways or routes that people commonly use, or places where people are commonly going, where there might be excess exposure, the way to do that is to understand what virus is being transmitted. So where are the infections coming from, and what are the epidemiological lines that you can follow? And the only way to really do that is to get the sequence of that virus. With the sequence, you can actually trace which individuals are in contact and infecting other individuals. You can understand what variants, we've all heard of variants, and we're all told to be afraid of them, but we shouldn't be afraid because, as far as we know, the vaccines are effective against all the known variants. But we still want to know if they're around. We want to know whether things are going to be scarier again, or what the major problems are. And the way to do that is to get these sequences. So how do we do that? In some countries, there were very effective virus sequencing programs. The United States lagged pretty far behind in most instances for effective virus sequencing and surveillance programs. But we, Isabel and David, who's going to speak later, mostly, were really very excited and adamant about bringing that possibility to our group. And could we really lead the way for the state of California and for the US more generally in generating the type of data that we're going to be useful in this way? And so how do we do that? We don't have a big sequencing core facility here on campus. We don't have a hospital, but we do have this amazing facility that Jeremy set up, and can we start there? And they reached out to me. I run a lab on campus called the Paleogenomics Lab. We are sequencing things like mammoths and mastodons and giant short-faced bears and even dirt to get DNA directly from dirt. But we had not been sequencing viruses. But crucially, what we did have was a sequencing machine, an Illumina sequencing machine on campus that we could use for whatever purpose we wanted to, because it's ours and we don't have to worry about asking permission from someone else. And so they brought us in and we had these big conversations about, oh, how are we possibly going to do this? It's going to take forever. We don't have the right path of actually bringing the virus samples in. And we managed to pull together an amazing group of people. Miten, who you'll hear from in a moment. There's a graduate student in my lab, Josh Kapp, who's been absolutely pivotal. In fact, he just sent, while Jeremy was talking, an email to a group of us saying that the next batch of sequences have come off the machine, and therefore it's Russ's turn to do work, as you'll see as we get through here. So Josh is always working, volunteering his time to put this together, and he and Miten have really been absolutely fundamental and amazing in making this happen. So the sequences get delivered to Miten. He spends a day turning them into something that he then transfers to Josh, who can turn them into a library, a genomic library that can be sequenced. And then we put that on the machine. That takes another day. And then the machine takes 36 hours to run, and it spits out somewhere between 96 and around 400 sequences, depending on how many different samples we give the machine. And then we send it off to Russ, who then does his magic that he'll tell you about and tells us what the sequences are. And then we can use that information to trace the evolutionary history of those viruses. We can say, oh, we have this number of this particular variant, and we have these individuals who seem to have been a transmission chain. They cluster together. And this is why getting the sequence is really important. Knowing that someone is positive, that we have a certain rate of positivity, is obviously really crucial for determining what our local response should be, as far as what sort of what we should do as far as going to grocery stores, having restaurants be open, et cetera. But if we have the data themselves, we can then better understand how to do surveillance. We can know what the future might look like by understanding more about the transmissibility of the different viruses, variants, and we can also help. So now the goal is, we have a very low infectivity rate here in Santa Cruz. We're really trying to help the state of California, really focusing on Central California. We're partnering with different public health institutions in California and perhaps beyond to be able to expand our capacity to do this sequencing and then to make this data accessible. And with that, I will end and pass it on to the next person.
Thank you, Beth. That was a perfect segue towards Miten. So Miten Jain is a research scientist in the UCSC Nanocore group. He's a leader in Nanocore sequencing development and projects. He's optimizing sequencing methods and developing software for MinION and Promethean nanopore devices, analyzing sequence data from bacterial and human epigenomes, and developing novel methods to detect regions of interest, as well as base modifications in genomic DNA and native RNA. He's here today to talk about how he's brought his expertise to virus sequencing. So you've heard a little bit about this from Beth. Equally, Miten will tell you what the nanopore can do for virus sequencing as well.
Thank you, Isabel. You've heard why sequencing is important, and you'll hear from Russ what sequencing does. There's a little bit of a connecting piece between when you have a SARS-CoV-2 positive sample, people who've been infected, and when we do sequencing. And that's kind of an important piece in that contamination is very important. So when we started embarking on this endeavor, it was really important to make sure that contamination does not happen, because that really can spoil the information that comes out, and the public health utility can severely get hindered, if not limited. In that regard, one thing that we ended up doing was we created an infrastructure to have different pieces of the workflow happen in different rooms with appropriate equipment and so on and so forth. Now, the key in all pandemics, as you'll hear from various epidemiologists, if there's one unique theme that comes along, which is rapid detection, rapid response. The MDL from the Genomics Institute allowed for rapid detection. We can turn around samples in a matter of hours when they arrive. The next part is rapid response, which is where the sequencing initiative comes in. At the moment, we can do, from when we get a sample, within a 3-day turnaround time, we can have full sequence information that Russ has communicated with County Health many times, as you'll hear from him. Having said that, there are times when you need even more urgent sequencing. We have a sample that was requested to Isabel by County Health today that we will be processing tonight, just because it's an urgent request. And as an institute, we are committed to community health and public health initiatives. So that will likely get turned around in less than 3 days' time. But we are still talking days, and it would be nice if we could do hours instead of days. And that's where nanocore sequencing comes in. And for people who do not know, nanocore sequencing is not only fast and portable, but also a UC Santa Cruz invention. Dave Deemer and Mark Akison are the inventors of nanocore sequencing, who are both faculty in our department in biomolecular engineering and members of the Genomics Institute. That being said, what we can do with nanocore sequencing is we can literally have, for a sample that arrives in the morning, we could do a diagnostic test, followed by the sequencing preparation, followed by sequencing, followed by actionable variant calling information that goes into Russ's hands, all in a span of 12 hours. And that becomes very powerful as a tool, because now you're talking information that's within a day turnaround time. Essentially, think of it as for travel at airports, or for urgent samples, for genomic contact tracing, for epidemiology, for strain surveillance. Anything that you can think of obviously benefits if we can increase the time, reduce the time that it takes to get actionable information. We are now essentially in process of completing our workflow, which is building up nanopore capacity to sequence on the order of tens to hundreds of samples on a regular basis. And what this does is that between our group and Beth's group, we have capacity to sequence hundreds of samples a day if there is a need for a public health initiative. And that we are hoping will contribute to the county health more and more, as it will with our collaborations that are about to start in terms of more samples from our neighboring counties, San Benito and Monterey, that you'll hear from David at the end, and other initiatives that we are part of, both with the US CDC as well as California Department of Public Health. And with that, I'll stop and hand it back to Isabel.
Thank you, Matan. A reminder to everybody, please do send in your questions. We will get to those as soon as our panelists have completed their talks. So our next speaker is Russ Corbett Dedic. He is an assistant professor of biomolecular engineering and an affiliate of the Genomics Institute. He studies the processes that govern genetic diversity in populations of species ranging from fruit flies to humans. His research combines classical experimental approaches in the laboratory with novel computational and statistical methods to analyze large genomic data sets. As such, this background really positioned him perfectly for his current work, which focuses on analyzing virus sequence information to better understand virus transmission and, importantly, identify the variants of interest. And he's been instrumental in the work that we're doing on virus sequencing. So off to you, Russ.
Sure. Thanks, Isabel. And of course, thank you, Beth and Matan, and well, Jeremy's gone, but thank you, Jeremy, as well, for setting up this topic so well and giving people a primer on what I'm going to talk about a little bit here. I'm going to talk about our work that we've done on sort of two levels. So you've heard a little bit about what we're doing in Santa Cruz, and I'll talk a little more about that and how we're using the tools we developed to help inform public health decisions in Santa Cruz. And then also a little bit about how our work has sort of a global impact as well. And the stuff we're doing is really the way, or we think it's the way to link up this information kind of across the entire world. And so to start with, I'll just say that, yeah, genome sequencing, like all these people, I'm convinced it's a very powerful way to respond to the pandemic. I think it's a really essential idea. And you know, Beth mentioned these variants, and I want to echo the point that you do not need to be scared of variants. I know that there's a lot of news out there that says, you know, variants are coming for you, and there's a double mutant now, and all these things that you hear. But we do need to track them, right? So the virus is evolving, and it's evolving to become more transmissible, so it's better at getting between humans. And in some cases, it's evolving to evade immunity. If somebody's been infected previously, it's acquiring mutations that allowed it to get around the immune response that that person's body has learned. And so what I'm often doing the minute I get sequence data from Mitane and Josh is I'm putting it into our software and I'm trying to figure out, well, what's in these data? What variants do we see? And what do we know about those variants from everywhere else in the world? And so often what we'll do is I'll stare at the data quickly and I'll just say, hey, there are these 3 or 4 we know about, pass that information on to public health and say, you probably, or we think that you should prioritize contact tracing efforts to look at these cases and understand how this may be spreading into our community and around. And of course, I don't know any more than that. I just know some anonymous sequences go somewhere. And that I think is the #1 thing people are starting to do with this technology now. And I think a really powerful thing that we're able to do. And I'm really proud to have been a part of this. So everyone's talked about speed, right? Jeremy does this. Jeremy's group figures out if you have a virus in hours, it becomes a genome in, I don't know, 18 hours, something like that, very, very quickly. And then we actually analyze the whole thing in tenths of a second. So that's what we've been doing, is building tools that are that fast, that can take this information and give us actionable results kind of as soon as I have them. And that kind of leads into the second part of what we've been doing, the kind of global reach of our work and why data sharing is so important. I think it's pretty obvious that the pandemic is a global phenomenon, right? It got everywhere in the world in less than a month. And it continues to spread between countries despite lockdowns, despite the fact that we've got sort of barriers and quarantines in place. It's getting around really, really quickly between all sorts of places. And so what we want to be able to do is to use this information and include it all in one place such that you can analyze it as a set, right? And so what that means is we need to be able to share this information and we need to be able to cross-reference this information. And the way, I'll talk a little bit about the cross-referencing. The idea is you need to be able to compare your virus genomes to see which ones are similar to others. And the way we do this is using a piece of software that we developed during the pandemic, actually. I'm extremely proud that we were able to build something that is in use now as a part of responding to the pandemic. And this allows us to compare sequences, like I said, in tenths of a second. So if we get a new sample in Santa Cruz, I can often tell you, hey, the closest relative that we know about in the world is over in the UK. So there's a good chance there's a travel history related to this sample. And that's the kind of very direct information we're getting out of these tools and approaches. Okay, and with that, I'll just turn it over to David, but very quickly to say that we're, right now, there's about 1.4 million viral genomes that have been shared and sequenced. Every day, 100,000 more pour in. And so, we're not done. I'm a little afraid that our tools won't keep up someday. So there's gonna be a lot more to do here. And we're really excited to keep working on that together. Okay, thanks.
Thank you, Russ. So you heard a little bit about David already. David Housler is the scientific director of the Genomics Institute. He's also a Howard Hughes Medical Institute investigator, a distinguished professor of biomolecular engineering here at UC Santa Cruz. His work explores the crossroads of molecular biology, mathematics, and computer science. His research has helped revolutionize the field of genomics with the introduction of advanced statistical and algorithmic models. His work accelerates the understanding of cancer, of human development, evolution, many other active areas of research. He's won zillion of awards, so I won't even try. And with that, I'm turning it over to you, David.
Let's unmute. Thanks so much. And thanks everybody for sticking with this. I am really, really honored to be with such a team. And I cannot say enough about how the people that you see here on this call and everybody that's worked with them has stepped up in this situation. It really is extraordinary. And I'm incredibly proud of how the Genomics Institute has responded to this situation. And it's not unlike how they have responded to previous situations. We've run into many circumstances that nobody could keep track of the cancer genomes, or maybe a private company would patent the human genome sequence and so forth. And so in the past, we've stepped up when needed, and I'm really, really proud this team has done it again and passed the test with flying colors. Everyone is clear right now that the world was not prepared for a pandemic. And that's not a technical problem as much as it is a social problem. However, there were some big technical issues, and Russ mentioned one. When we tried to do comparative analysis of all of the SARS-CoV-2 genomes, the virus that creates COVID-19, all of the current software broke. And it was just amazing to see Russ's team come in and build an entirely new suite of software on the fly, in the moment, and allow us to accomplish this. So there were technical challenges. There are challenges that Miten is facing on how to get the sequences really fast from the nanopore. And this hadn't completely been solved before. So we have these technical challenges that are overcome. And by the brilliance of this team, many of them are being overcome. But there have also been serious social challenges. And everybody has to understand that the sequence of the virus itself, the A's, C's, G's, and U's of the RNA of the virus, is not personal information. And we should be able to share the information we read from the virus genome itself immediately and globally. There have nevertheless been a number of obstacles that we have fought hard to overcome with this. Part of it is local public health was simply not prepared to generate this data and share it. And we are really working with them to network this all the way from local to state to national to international, as you heard. Really, we can have global situational awareness of everything the virus is doing at any given point, all the way down to the molecules in the virus, the message that the virus has, and its strategies for trying to get around our defenses. Now, the virus has not succeeded yet in creating a new strain that is not responsive to our vaccines. Our vaccines are still holding fine for the existing strains, but that does not mean that this won't happen in the future. It does for the flu, for example, happen that you do get a new strain and you do need to be re-vaccinated against the new strain. So we need to be prepared for the possibility that that will happen someday with COVID. The only way we will be prepared is to have this global situational awareness of everything the virus is doing at any given point, from the point of the genome of the virus. The other part of this is what Beth was talking about, how the virus spreads from one person to another. And it's essential that we be able to track the molecular path of the virus and to be able to compare that information. Now, here's where some people get uncomfortable, and we do need to protect personal privacy at this point. Our job at the Genomics Institute is to track the molecular information about the virus. Our job is not to track personal information about members of our community. That is the job of the authorized personnel at the local Department of Public Health, who are trained to carefully handle this information. So we have a strict separation between the information that is available to trained public health officials and the information that is available to us. And that's a good thing. But we also have to be able to have a smooth communication, and we have to have a rapid and effective communication so that we can put in the hands of the officials who can do something about it. We have to put in their hands all the information they need to be able to track the virus. And we're now building nice tools that are like Google Maps, which essentially show you the different variants of the virus and how they are being spread around the community, and details from the molecular point of view that can be matched with other information. And this occurs in the office of the local public health. And we want to do this actually for California as a whole, and for the nation, and for the world. And I think we have an opportunity to work together to build this kind of last mile of the epidemiological investigation software, where we actually bring the information into use while still vigorously protecting personal information and privacy of our citizens. So this is a great challenge. I want to make sure that everybody is excited about it, that you're thinking about contributing to it if you can. we will make a huge difference. We do not want to be ever again blind to what is happening with infectious diseases around us. With genomics, we can know what the infectious agent is doing at every turn. And there's no excuse to ever going back to a situation where surveillance is simply ignored or impossible or beyond our means. If we set up appropriate surveillance of pathogens now, we will be ready for the future of this virus and for all other viruses. So thank you so much for listening to all of this, and do contact us if you think you can help.
Thank you, David. We have a first question that came in that is multi-layered, and it's very important because it talks a little bit about where we were situated at UC Santa Cruz to start this work. So I'm going to read the question, and then I'm going to dissect it a little bit. So regarding the community virus tracking, I have a hard time imagining a university without a medical school being able to set up this virus detection and virus sequencing. How is UC Santa Cruz able to rapidly move resources and expertise across departments and divisions? How did senior administration help move this along? I'm wondering if we could start, David, maybe with you talking about the vision for UC Santa Cruz and in particularly the Genomics Institute around responding to health issues and precision medicine approaches in particular without a medical school?
That's a great question. We've been able to do remarkable things at the Genomics Institute without a medical school. And the reason is that increasingly medicine depends on information, and it depends on the ability to do molecular tests that are a window into what is going on. So the combination of sharing information, doing molecular tests, comparing molecular tests to other data banks of information, all of these things are things that we can do really well. And we've demonstrated that we are actually world leaders when it comes to putting the information from the molecular test of a single individual into the context of the global information about that molecular test. And we really cut our teeth on that in cancer. And I want to point out the important contributions of Elena Vasque, who is our endowed chair of pediatric genomics. Her work inspired many at Santa Cruz to participate in a project called the Treehouse Cancer Project, where we actually engage in about a dozen projects with researchers at hospitals and medical institutions around the world. And these include Sanford, UCSF, several other places in California where difficult to treat childhood cancer cases are faced by doctors, and they need to know, how is my case similar to all the other cases that have ever been seen anywhere in the world? And can you give me some information that might help the treatment of this particular child? And in fact, the answer is yes. And Elena's team has come up in many, many cases with life-saving information. They look at about 15% of the cases of untreatable or difficult to treat cancer in California every year now through the Treehouse. And they provide genetic and molecular advice on how this case could be approached above and beyond the standard of care. And in that, we have Elena's training. For example, she is trained to run a diagnostic laboratory. She knows all of the legal and regulatory requirements of running a diagnostic laboratory. We took advantage of that to launch the Molecular Diagnostics Laboratory. And our leadership is very, very supportive of this from day one. They made sure that we had the resources we need, the larger we, the people you've heard from here, including Jeremy and many others, that we've had those resources to build on the early clinical experience we had in cancer in other ways and rapidly adapt it to treating the issues that came up with the SARS-CoV-2 virus.
Thank you. So the Genomics Institute is actually cross-divisional, and one of our core values is really incorporating research from different individuals, different areas of expertise together to move forward genomics research. And so here on this panel, we actually have 2 different divisions represented in 3 different departments. So part of this question really is about the collaboration across departments and divisions. I wonder, Beth, if you could talk a little bit about that from your perspective as you've been working with the Genomics Institute and are in a different division, of course, and department than David and Russ and Matane.
Sorry, talk about the collaboration?
Collaboration across divisions and departments. So one of the questions here was how are we able to quickly mobilize resources and expertise across divisions and departments? And the way you integrate across departments and divisions, I think, has been a big piece of that. In addition, you talked a little bit about how you were able to help quickly with this effort by using your sequencer, which was there for other purposes, right? It was repurposed for this.
I guess, you know, when I read this question, because I was reading the questions as they were coming in, my initial response to this is that not having a medical school or not having a hospital might actually have made it easier for us to do this. We didn't really have the administrative burden that would have been associated with starting something like this if we'd been associated with a large medical school. And in a sense, because it was something that was brand new, there wasn't already a whole bunch of bureaucratic red tape that we needed to tear through and try to figure out how to fix and finagle in a way that was going to make it work. Instead, we have this institute, the Genomics Institute, is relatively new and definitely really aiming to create opportunities for collaboration across divisions within the university. And it was kind of ideal in a way that you don't like to say because you don't want to jinx anything, right? And that we really were able to just turn around and say, well, okay, you know, we've been kicked out of our labs. Basically, we're not allowed to do anything because of the situation, because of the pandemic, unless we're focusing on doing something about the pandemic. So here really is an opportunity where we have all of these scientists who are desperate to contribute in some way, who have quite a lot of downtime that they didn't want to have or plan to have. And here is a way that we can bring all of our expertise together. And, you know, because of the experience that we've had with cancer and with the human genome and all of these projects that really capitalize on expertise in molecular biology and also genomics and bioinformatics, we just had the coalescence of the right people at the right time to be able to pull this off. And I really think, you know, rather than it be a burden that we don't have a medical school and a hospital, it was a bit of a blessing. We were really just able to jump in feet first and really just hit the ground running. I mean, it would have been better if we had tons of money and tons of infrastructure and larger sequencers and more people. But as we are, our small university, UC Santa Cruz, yet positioned with this incredible international expertise in bioinformatics and sequencing and processing and thinking about these things, we were just positioned in the right way to make this happen. And I think this is just an amazing benefit of the existence of the Genomics Institute overall. I mean, really, it was crucial to being able to facilitate this interdivisional transfer of expertise and communication and kind of circumvent the bureaucratic red tape that might have gotten in our way. As it is, all of the bureaucracy that we encountered, as David pointed out, was from existing medical institutions that didn't really know what to do with our ability to generate data that were completely not associated with any individuals and yet an incredibly useful resource. And it took a long time to get it through to people that, yes, this is just virus data. There's nothing about this that's identifiable as a particular individual. We can do with this everything that we want. The lawyers have all had a look at this, many looks, in fact. too many looks, perhaps, for some of us that were in there. What's going on. But it was really just a tremendous coalescence of opportunities that I think was actually aided by our being outside of a medical institution rather than thwarted by it.
I would like to add quickly that you would be amazed how broken the relationship is between medical institutions and public health. It is badly broken in this country, and there is a huge amount of bureaucratic barrier and just misunderstanding and mistrust. You know, we being a neutral party, not already being entrenched in a medical school viewpoint, did help. I agree with Beth.
And there's a record at UCSC of working with medical institutions in partnership to move forward efforts. And I was thinking as we were talking about what we can do without a medical institution and how in some ways it's a benefit. Matane, I'm wondering if you wanted to talk a little bit about your project with the Nanocore on undiagnosed diseases and let people know the advancements that are happening in that space.
Sure, and I'll quickly just piggybacking off of what David said and what Beth was saying, despite not being a medical institution, the challenges were more on the regulatory side. Our institute, essentially through a multidisciplinary, multi-department collaboration, established a diagnostics lab within a month. May 1st, the lab was there. We've had sequencing capacity since July of 2020, so we could sequence in July. The delay was not at the Santa Cruz, and it was what David and Isabel were dealing with, the healthcare providers and the clinics around. So it's at the university, it was actually easy to make things work. It was the extraneous elements that are beyond our control that our leadership had to deal with. And in a similar way, we've been collaborating with both UCLA and UCSF on clinically relevant undiagnosed disease programs. The idea being, there are these pediatric diseases, for example, that do not have causes or causality defined. The genome sequencing looks okay for the kind of genome sequencing that hospitals are used to doing using short reads. Also looking at the exome sequencing data. Exome sequencing, for those of you who do not know, it's essentially looking at the parts of your genome that code or that correspond to genes that express and turn on and off and those things. So these are diseases that obviously are present in these kids, but their causality is unknown. And what we are addressing in collaboration with these institutions is to look at long read sequencing data using the Nanocore, because we can get very long reads along with information that includes these epigenetic marks or these these features on the genomic DNA that essentially dictate if a gene is turned on or off in very simple manner. And what that allows you to do is to look at these effects of why a particular genome that looks otherwise normal or nothing odd with it corresponds to a causality of a disease. We have a case at the moment that we are working on, for example, with UCLA on a kid with pediatric Wilms tumor. And for all intents and reasons, nothing's wrong when you look at short read based genomes. The exome sequencing, the gene coding bits of the genome look fine. When we look at Nanocore, there are some oddities, and we are investigating into what can we learn by applying these long read sequencing methods to this kind of a problem. We have similar collaborations in looking at pediatric leukemia, where chromosomal translocation, so something happens to a chromosome and two chromosomes mix in a bad way, and what its technical term is called is Robertsonian translocation. And when this odd thing happens, the risk of cancer in children 8 years and younger is increased 5,000-fold. And conventional sequencing does not have an answer to even what's going wrong. But long read sequencing starts affording a look into what's happening in the genome. And let's look at just that genome itself. And then we can also follow it up with looking at the genes themselves, both in the DNA space, their RNA transcriptome space, the transcripts that are actually making proteins, and then the multi-omics assessment of those. And the goal in the longer run is that in an ideal world, we'd be sequencing every infant that's born in the state of California, or ideally in our country. And we have a snapshot of what that genome looked like at birth. So we can help over time when, God forbid, things go wrong with children or individuals. Ideally, we are truly embarking on what will look like in terms of precision medicine and personalized genomics. And it's not a term that's a buzzword anymore. it's reality. There are projects across the country and across the world that are starting to use these genomics-based methods, using a lot of our expertise, both in terms of the laboratory side with technologies and on the informatics side with the software. Least of all is the genome browser, where any and everything that we do in the informatics at some point or another crosses the UCSC genome browser. So going back to the vision back in 2000 that David and Jim Kent had of creating a resource that serves its purpose to humanity for ultimately what will be a clinical genomics endeavor.
Thank you. So there's a question here around how we manage collaborations with industry and government facilities like the NIH, CDC. And of course, this matters because this work that we're talking about is only as impactful as it reaches the people, right? And it integrates with other agencies that are critically involved. Russ, I'm wondering if you can talk a little bit about the work you've been doing in facilitating those collaborations and really making sure that our work is integrated with larger efforts.
Sure, yeah. So as Isabel said, this is only any good for anyone if we get the information out. And so what we do, first of all, we develop all software in open source formats and we distribute it. So anyone in the world can download it and run it. And we actually work really, really hard to make that as straightforward as possible for everyone to do. So it's a easy process to get our software and try it. And with that, so the number one thing we did in this pandemic is a software package David and I talked about a little that has simplified and dramatically accelerated the analysis that needs to go on here. And we, I was fortunate enough to be able to reach out to the CDC. And in fact, if you look at their genomic epidemiology toolkit, they made a series of modules for how to use this information in public health offices. I'm actually, one of the fourth module that was recorded was mine. So you'll see my face telling you how to use our software and what exactly it does. The other thing we've done with this, though, is we recognize that public health is simultaneously centralized, for example, for the CDC, where they have lots of information coming into one place, but it's also extremely distributed, right? There's little offices all over the world doing local public health. And so the other main thing we did in collaboration with the Genome Browser team was to make this package something we would run for you. So you would give us your genome sequence via the internet, and we'll tell you kind of everything we think you need to know about it very, very quickly. And so that's kind of the idea, is that we make the software available and we even run it for you to make it straightforward to get. And then with respect to integration, so we've recorded videos for the CDC, other large scale agencies. I've applied for funding from the NIH, and my program officer on my current grant has said it's fine to repurpose the funds to work on COVID. So I've done that. And I think many of us probably have done that. I assume all of us, yes. And so, those agencies have been extremely supportive of this work, and we're really excited to continue to work with them to develop these tools, to build them into CDC modules and NCBI modules, and also to hopefully get more funding to keep innovating and building new tools to do this kind of stuff for the next steps.
I would add to that quickly that we are integrators and networkers. We enjoy working with a number of constituencies because we're all about data sharing. We've gotten a lot of experience in networking a number of institutions to get them to work together. And I must say that there are a lot of big name institutions, medical institutions, that don't really work well with other institutions. Most of the big projects that are planned for NIH, we find that one institution wants to do it all and doesn't really want to work with partners. We find that the different NIH institutes for different diseases don't work together. And of course, the NIH working with the CDC, well, that's really a difficult call. But not being an established big shot elite institution actually helps us here. And some people say because we're not threatening them. So we can be the glue, the networking that pulls together your household top name elite institutions into actually working together and actually sharing things and actually doing something, not just all controlled by their own institute, but sharing. So I think that's something we've been better and better at over the years.
Thank you. We have an important question here about what happens in the years to come, right? So we talked a little bit about how we're trying to create a structure here that will be useful post-pandemic. And the question is, I imagine the data set could be mined for years to come. What plans are there to continue to leverage the infrastructure and data analysis capabilities after the pandemic is over?
That's Russ.
Well, I certainly have ideas about this. I've been thinking a lot about what I think the future of what I'm calling genomic contact tracing holds. How are we going to use this information? So this touches on sort of two different things here. This specific data set from this pandemic, it's going to grow at the end. Well, who knows what the end will be, but in the near future, there will be 5 to 10 million viral genome sequences, which is an unprecedented pathogen sequencing effort. I think the largest data set before that was maybe a couple 100,000 bacteria of some kind. So it's orders of magnitude more. And the kind of evolutionary mysteries and thinking about how the virus has evolved within human populations, it will be a subject of study for the next decade or decades among many different groups, is figuring out how we can learn the most out of these data so that the next time something like this happens, we're a little bit more prepared and it doesn't immediately break everything that we've been doing 3 months into the event or whenever that happened. It was fast. We were not ready. And so that's going to be huge. With respect to what this does outside of that, so this is much more general than a pandemic. The exact same sort of principles that we're talking about are going to work for foodborne pathogens. So for example, you see a new bacterial strain that's making people sick. We'll do the exact same kinds of analysis with important changes to match those needs to figure out where it's come from, what we know about that particular pathogen, and how we can deal with this. And as we get more and more precise, it's going to touch on things like what Matane is talking about, how we can use personalized medicine. Well, we're going to have personalized pathogen genomics as well, where I know something about that very specific strain of this pathogen, and it may influence treatment outcomes. For example, if we can get that information fast enough, a doctor might say, we know this strain of the virus is better treated by some other drug or some other mechanism than other strains of the virus. And that kind of information, we've really been stressing this speed of turnaround, how quickly we can leverage all this information. That's going to be the big revolution in pathogen genomics, is people developing faster and better ways to use this information, to integrate this information, and make intelligent, informed decisions about how to use it on the fly.
Thank you. One of the things that is very much true about this effort is that there have been a number of challenges along the way, and some of them we've talked about. Beth, I'm wondering if you can talk about some of the barriers to getting this sequencing going in the first place, and sort of walk us through what your story was like in terms of joining this team and trying to make it happen so quickly.
Well, I mean, I guess there are two different layers of barriers, and one of them is just the sort of legal and administrative hurdles of trying to communicate through the university's lawyers about what the risk of providing this sort of information is, and with public health. And the other is infrastructure. And turns out that while we thought the first was going to be the hardest problem to solve, the second really has been quite challenging since we've gotten through. We've had some really good luck. We're trying to provide this service to the county at no cost to them, so we're trying to absorb all the cost ourselves, and most of the scientists who've been involved have been volunteering, donating our time for this. So my graduate students have been working on this, my postdocs have been working on this, people in Mark Akeson's group and Miten's group have been volunteering their time to work on this. We've been fortunate, we've had some donations from alums, so thank you very much if you're listening, alums who've helped us out to get this started. We've been able to bring in a couple of robots recently. In fact, only one of them is working, and it was installed 2 days ago, and the second one had a faulty part, so it will be installed, and that will help us to speed things up. So now we're really at this junction point where we would like to be able to do more. We would like to be able to scale up to really make this something that we can do, not just for the current pandemic and for where this is going to go, but for future pandemics. We have this fantastic infrastructure, really great enthusiasm, and absolutely the right expertise without any links to any other institutions that would necessarily hold us back or actually push us in any different direction, which makes us uniquely positioned to be able to provide this sort of resource to the community locally statewide, nationally, and even internationally, but we need to be able to scale up. So what do we need to scale up? We probably need a larger sequencing machine. We definitely need personnel. And this is really where the biggest barriers have been right now. It would be amazing to be able to bring in one person to work in Miten and one person to work with me and one person to work with Russ, who could really just be focused on this, and not necessarily just COVID, but what can we do for other circulating infectious diseases that we might be able to apply these new technologies that we've learned in this time of the coronavirus to other things that are potentially circulating. We know that, the more we push into places that haven't had people in them, the more likely it is that we're going to see additional infectious diseases and an increasing burden of epidemics and potential pandemics in our future. And we can be prepared locally and nationally and internationally to be able to have a response to this. We are in a position where, because of who and where we are, we could position UC Santa Cruz and the Genomics Institute as global leaders in this, but we need to grow, and we need the infrastructure, the support, the place to be able to do this. And right now, like the COVID lab is sequestered part of my lab that we normally use for ancient DNA, sedimentary ancient DNA sequencing. And those people have been kicked out for the time being to do this particular lab. We have a brilliant facility that I can see out my window, so I focus down there on Delaware, that we could move into, but it is not outfitted as a lab. And this is expensive stuff that nobody cares about, in the institution anyway, about helping us to outfit. And we would love to be able to create the resource, the lab down there that we could use for this and for the future work that we're doing. But that requires raising the money. So that has really been where the bottlenecks have been right now. And I mean, it's an exciting time, and we really are making incredible strides doing things, piecemeal and with volunteer time. And we're going to continue to do that because we are making an impact and we are giving people data that are useful to them that they could need. But this is also an opportunity for us to capitalize on who we are, on our unique identity, on our unique positioning, and use this as an opportunity to make Santa Cruz a place that is known for delivering exactly this. And to do that, we have to grow. So we need to raise some funds for that. I don't think you wanted me to say that, but this was the right time for me.
So true. No, it's absolutely true. And to your point about alums having donated money, yes, that is absolutely true. And it's made it possible for us to really jump in and do this work quickly. And Miten, you have been able to hire somebody to help out to get this going in your lab as well. Is that right?
Yes, we've just extended an offer to somebody who will come and take on a part of this workflow full time, and to essentially do what all David, Russ, and Beth have been talking about, that this pandemic is the prototype for setting up the infrastructure for, it's not a matter of if, but when the next time comes, we are ready for that rapid detection and rapid response. And we also, like all of us, have collaborations that are extending into the, what is considered, as people might be recalling from news, is the host genome interaction. When somebody's infected, what happens to their human aspect of the genome or the human microbiome? So analyzing and understanding those in a whole genome perspective, again, kind of making the full picture so that it's, as Russ said, it's personalized pathogen medicine, because that is eventually going to be a part of the personalized medicine initiative. It's not, we always think of personalized medicine as the CAR-T therapy and the cancer part of it, but this is the non-glossy aspect of it, which actually is important to a lot of the people in the community over time.
We have an extraordinary opportunity in doing DNA and RNA analysis on the human immune response. And that, I think, will be a big part of the next wave. We'll see a lot of research over that. We'll see a lot of progress. As many know, cancer is now treated most successfully with what are called immunotherapies. And they beef up the cancer patient's immune system to fight off the cancer. So understanding the immune system and being able to modify the immune system to increase its power to fight disease, including cancer and infectious disease, is a big task for genomics in the future. And we're really well positioned to play a big role in that with all of our infrastructure.
Thank you. That leads into another question we have. So I would like to know if the viral genome that is built is related to epigenetic factors, meaning that is the viral genetic map that is used by public authorities linked with epigenetic factors. Does this affect the treatment option approach in the future?
Let me just say quickly, because that addresses what we were just talking about. If you want to understand the host, the person who is infected, if you want to understand how they're reacting to the virus, then you have to understand their immune system. And to understand their immune system, there are things that we can do in the future which are not routine now, but we can actually look at their immune cells that are mounted in defense. These are what are called B cells and T cells. The B cells make antibodies and the T cells make receptors that recognize the virus and raise the alarm. Each one has a slightly different structure. So we're again in the fascinating thing of large-scale sequencing. Right now, there are millions of T cells circulating around in your blood system with slightly different receptors, and your body is constantly making new ones. But when one recognizes a dangerous new pathogen, it sets off the alarm and starts making new copies of itself and specializing further to refine its ability to react to that. And then when it's finished, it will create what's called a memory T cell, so that if you ever see something like that again, you will rapidly react to it. This is a great frontier for genomics, and there's exciting new work on actually sequencing the T cell receptors and understanding how an infected person is responding, and coupling that with total analysis of the antibodies that this person is making from their B cells. This is great. We should be able to do this. We're also being able to test how the virus reacts in different tissues. Many labs are growing what are called organoids, small pieces of tissue that are very, very similar to human lung tissue. They're actually grown from human cells into human lung tissue. And then we can do experiments on how they are reacting to the virus. And these technologies, the ability to grow organoids, is another big research project that we have at the Genomics Institute. So immunology, organoid research, all of these things can come into play in the next wave of activity. And what's going on here is, to a certain extent, epigenetic. It's epigenetic to the virus, but it is certainly genetic in the larger picture as well.
Thank you. Can we talk a little bit about going back to the sequencing? What is the quantity of sequences that you need to have valuable, useful information locally? Where does the point that we need to get to be able to have an impact for local health efforts? Russ, do you want to speak to that?
Sure, yeah. So nobody knows, but the answer is more. And I do mean that exactly as I'm saying that, which is to say that it's extremely hard to design a specific sampling scheme that we know isn't going to run into problems. And the truth is, it's not that costly to sequence these. Obviously, it takes people time, it takes effort and energy, but the actual cost of the virus itself is not that much. And I'm of the opinion that the more we do, the better off we are, the more we know. The more subtle answer to this question is, it depends on your goals. So if you're doing what's called viral surveillance, which is basically trying to ask the question of how many of a given strain of the virus is in this area, what frequency is that? So for example, you've heard of the UK variant or things like this. How many of those are here in Santa Cruz? Most people think you need somewhere in the range of 2 to 5% to do that reasonably well. If you're trying to do the thing we're talking about, which is what I think is in most ways the future of how we'll use pathogen genomics, that's genomic contact tracing. That is saying that we, a public health officer that knows the identities of these people could figure out that this person transmitted to this one, indicating that there is some connection between these cases. Then you need to sequence them all, is the answer. You really want to have all of that information. And I think that will be the future we're going to build toward. I think the capacity is being built up in many public health offices across the country and across the world. And so the not so distant future will be exactly what I'm describing.
Thank you. And then for everybody who's listening, we touched on this earlier, but just as a reminder, so our COVID testing lab on campus, all the positive samples are now going to sequencing. But in addition to that, we are also getting separate samples from local public health authorities and testing those. And the extra samples that we are getting are samples that have particular concerns. Right, the symptoms are, for whatever reason, different than expected. The trajectory of the illness has been different than an average COVID illness. And so there's a reason that the public health authority then has become interested in the sequencing information. And that's part of the large value that can be provided by our local and very quick sequencing efforts. Beth, there's a question directly to you. It says, Dr. Shapiro alluded to the fact that UCSC could position itself as a global leader in infectious diseases analysis. However, we must grow first. So what do you mean by growing? Does growing primarily mean collaborating with large COVID testing facilities, or internally growing the personnel involved in these projects?
It's internally growing. We already have the collaborations with the large infectious disease centers. We have collaborations that are ongoing with CDC. We're sitting in on these phone calls every week that are not always that entertaining, right? And also with CDPH and the other public health departments. So we are among the players in this space who are doing these things. We also have unique expertise, which comes to us from our experience running the UCSC genome browser, our experience working with cancer, with lots of different medical institutions that aren't necessarily aligned with any particular university, that make us uniquely positioned to be able to do something different than these other big places can do. What we don't have is the actual logistical infrastructure, the space. We do have a space. We have a beautiful empty building. It's like a blank slate with which we could do whatever we wanted to do, and we know what we want to do and what we have to do. We just have to convince people to pay for it. So how do we do that? I don't know, but we're working on it, right? I mean, this is what David and Isabel spend a lot of their time doing, is figuring out what the best way to turn this into the facility that we want it to be. And there's a lot of potential there. You know, I don't know, do I personally want all of that beautiful space down there to be turned into infectious disease? No, I think it'd be great to have a world-class ancient DNA facility down there. You know? But that's what I mean. Really, the infrastructure, we have to create a lab, a diagnostic lab, out of this beautiful space that wasn't used for that. It's a giant empty warehouse that is ready for us. And the sort of some of the infrastructure is there, the electrical infrastructure, the IT infrastructure, all of that exists. We need the actual benches, the wet labs, the sinks, and the other boring stuff that creates a lab. And we need to build the human resources necessary to do that kind of thing. But in the absence of that, which is a pretty big stumbling block, but a silly stumbling block, right? Then we are well positioned to become global leaders in this space.
And I'll just add that we are leaders from the Genomics Institute perspective in having the technical know-how and the toolkit, the software and the technology. We have that. We are not starting from scratch. We are scaling things as is. We went from nothing to hundreds of samples a day in essentially a matter of weeks. So it's what Beth is talking about is from that into a very large scale production and service contributor, which we can do because we have both individuals who can lead that initiative and the know-how, both at the lab level and the software level. But it's the resources that we are missing on at the moment.
More than 10,000 human geneticists used our UCSC genome browser today. I'm confident of that because it happens every day of the week. And that is a huge impact on genetics. So we are already a world leader. We are, I think, without question, the most popular site on the internet to come to learn about human genetics. And our SARS-CoV-2 genome browser is picking up rapidly too. I think it's the third most popular site on our suite of sites. I think people, last I checked, right, it was the human browser, the mouse genome browser, because there are many mouse researchers, and then SARS-CoV-2 was the third most popular place people come to at Santa Cruz to look at genomes. So we are world famous for genomes already.
And to Beth's comment about the building, the big building that we have, we do have a question that came in here from an alum who was noting that when I graduated from the biomed building had just been completed. Where is the Genomics Institute? So for those of you that don't know, and are alumni of UC Santa Cruz, about 2 years ago, the Genomics Institute moved down to 2300 Delaware. And so we still have a presence on main campus because we have professors, of course, across divisions and departments who are teaching. And so they have offices on the main campus. But we, as the Genomics Institute, have moved into 2300 Delaware, and we have a large suite of rooms there where we're able to house that growing number of researchers that I mentioned to you earlier. So again, we're at 110 in terms of people that work with the Genomics Institute directly, and another 20 to 40 that are affiliates with us and are able to have hotel spaces there. And what Beth was also referring to is that there is a large building in the back which has all of the laboratory areas, and that is currently being built. And so the COVID testing lab was originally, when we started, because we started in 6-7 weeks, basically, from planning to the first test that we had. We were housed in main campus in the Sinsheimer building, having taken over research labs. And over the period of testing, because the number of tests that we received were just so large, right? And now, as Jeremy noted, we're over 100,000 tests. We moved to a newly built building in the back of 2300 Delaware area. So we have a brand new diagnostic lab, which another question that came in was about our diagnostic intents for the future as well. And that COVID testing lab is now, because we've learned how to run a diagnostic testing lab and all the regulatory requirements around it, that will transform into a genomics diagnostic testing lab in the near future. David, do you want to talk a little bit about the live cell biotechnology plans as well for that space?
Well, the other thing we're doing is creating a lab that's very different from your standard molecular biology benchtop lab. Usually in molecular biology labs, you set up the benches and then you buy a lot of very expensive equipment. For example, your microscope might cost $100,000 or $200,000, et cetera, et cetera. It's all very specialized, and each manufacturer tries to create a self-contained system that does one thing in your laboratory. We have a completely different vision of the modern laboratory. We are actually 3D printing most of the components that we use on our benchtop. Our microscopes are made from cell phone cameras, and we use little Raspberry Pi computers, $20 a shot. And basically that image that you can take on actual consumer CMOS surfaces is getting better, just like it used to be Kodak film and now it's all digital. The rate at which commodity enterprises that involve sensing and manipulating and building are emerging is absolutely stunning. So we have a microscope that we built for $85 a piece called the Picroscope, and you can use it with the Raspberry Pis, and it sits with our samples as our tissues grow. It actually sends up information to the Internet of Things 24 hours a day. You can use it from your cell phone at home. And a matter of fact, we used it in an experiment recently with high school students at Alisal High School in Salinas nearby. They were greatly inconvenienced by COVID. They couldn't come in to take AP biology. we set it up so they could actually use our microscopes from their cell phones, and they experimented with zebrafish growing embryos, and they treated them with different fertilizer runoffs, ingredients like ammonium nitrate. They found out that what levels actually affected the embryonic development, and they could do this all remotely through the technology that we built. Essentially, the Internet of Things is happening now. Devices are becoming smart and connected. Many of you may have some of these devices in your home. The ancient kind of laboratory equipment we're dealing with now doesn't make sense anymore. And in this case, actually, the way high schools teach AP biology doesn't make sense anymore. When we talk to the teachers, they said, yeah, we have to buy a microscope for every kid, and we keep it in a shelf, and then we have to hope that we can get the zebrafish embryos shipped to us and they don't die, or they're not improperly fertilized, and et cetera, et cetera. And it was, I can't describe the excitement. They said, well, no, we just have the students get on their cell phone, and you can actually do experiments over the Internet of Things. You can control the device, or you can say, give it some more ammonium nitrate. That's an extraordinary experience for a high school student, but it's an extraordinary experience for our team now, actually being able to open up the lab, fully connected to the Internet of Things, to be able to build cheap devices that actually do the job. And they do this at scale. So the other thing that I've found in being in a molecular biology lab is that because the equipment is so expensive and the whole technologies are so cumbersome, you end up saying, well, maybe we could look at 5 samples, or maybe we could get 6. That should be 600 or 6,000 different experiments that are done. And if you build technology that's smaller, faster, cheaper, more modular, and then actually make it so that it can be shared and it can grow into a new way of doing experiments. We're thinking of not only open source, but open source software, but open source 3D printing, where people can iterate and help on a design. How do you hold the cells? How do you provide media to them? How do you image them with a cell phone camera? All of this stuff is recipes online that are shared now, and I think will become popular in labs all around the world. So this is the second part of genomics. Really, it's getting the data from living cells in tissues into the computers. That's the key. And we have some really revolutionary ideas that we are playing out now in what we call live cell biotechnology. And it's a revolution in making new kinds of equipment that is much more like open source software, very modular, more like you would see in computer science than you see in the old-fashioned laboratory equipment. And I think we can really make a difference there.
Thank you. It touches nicely on a question we got just about really the idea of rapid change and opening up of how to address pandemics with low-cost nanopore sequencers, and then personal sequencers with DIY sequencing that really takes a new approach. I'm wondering, Matane, can you tell people quickly how, for instance, the MinION could be useful in developing countries?
Sure. So the MinION that Isabel is referring to is kind of a small table-sized stapler. So that's the size of that device. It fits in the palm of your hand. It's smaller than essentially the same size as an iPhone, and it weighs 90 grams. It's a portable DNA sequencer that can work in field with minimal resources, can sequence and produce data real time. So essentially, from when you click go, the amount of data that starts pouring out is a matter of seconds, and you can start getting actionable information within a matter of seconds, depending on the experiments one is doing. If you are doing a pathogenic bacterial detection in a sample from a hospital, the answer will start coming in as little as 15 seconds from when the sequencing starts. It's portable enough that it can be carried in your backpack wherever you go, works on a standard laptop. And the good thing is that it provides what we and the community call democratization of sequencing. Anybody can sequence anywhere in a simple enough manner. And this is simple enough that astronauts on the International Space Station can do this. There's other extreme environments on our planet, Antarctica, Alaska, and several others where Beth's group goes and does really funny sampling. The amazing pictures when you see these guys sample those things. And imagine being able to sequence right on site. And there have been amazing, real personalized applications where, for example, there's community who've used this portable sequencing inside an operating theater to do a real-time assessment of, have I removed enough cancerous tissue from a patient? And when I have, I'll stop the surgery. So you don't just go a little bit aggressively and remove healthy tissue. You can really do a very fine-tuned analysis of what can be done. And this is happening on the operation table in the theater, real time. That's what this kind of device allows you to do. And the nice thing is it comes in a variety of forms. So there is that small device that goes on the space station in zero gravity and works just fine. And there is the large Hulk-sized version of it, which can do hundreds of genomes in a matter of hours every day. So it gives you that range, but at the same time, in field to what would be a large-scale sequencing endeavor in a conventional laboratory that we have at the Institute. And that's why we think it is one of the key players as our institute goes forward in becoming a high-scale data production center.
Thank you very much. I'm conscious that we're coming to 4 and we have a beautiful question that's come in that I want to make sure is addressed. So it says, I'm a City of Santa Cruz community member. Would you expand on how you worked at the very beginning with the county, public health, nonprofits, community foundation? And then I hope the cooperation would continue. Does that seem like it might happen? So I'm going to give a try in answering that one. What I will say that has been absolutely incredible since the very beginning of this pandemic is that all of the researchers, all the staff members that have been involved in this work and addressing the virus have been committed to community impact. And so key to everything that we have done has been to understand what has been needed in the community by public health, by the clinics, and to work with them to move that forward. We also had an extraordinary relationship with the Community Foundation that has supported testing in Santa Cruz, support for people who have suffered from COVID in Santa Cruz, and came together and forward really to bring people into a network so that clinics were talking to each other, doctors were talking to each other, public health officials were talking to each other, researchers were doing that. And we had a campus administration, which frankly rose very quickly to the challenge and supported all the efforts of the people you're seeing on the panel and many others on the campus to get this done. And to the question about whether cooperation would continue, I'm very proud to say that Salud para la Gente just submitted a paper to an international medical conference talking about the collaboration in Santa Cruz. They have applied to this conference for 10 years running, their community clinic here, and have not had a paper accepted. Their paper about the collaboration in Santa Cruz was just accepted, and they will be talking about the importance of an integrated approach to responding to public health issues and the need for researchers to be part of it, for academic institutions to be part of it, for organizations like the Community Foundation that can bring all of these pieces together and the grassroots efforts that are happening in town together. And so that is critical to not only what has happened in our success, but also going forward. And we've built relationships that I believe are going to survive for the long term and be to the benefit of all. So with that, it is 4 o'clock. Thank you to our panelists. Thank you to all that have attended. We are very grateful and we look forward to continuing this work and continuing working with you and telling you about our work. Thank you.
Thanks, everybody.
You.
Thank you.
Thank you.